Welcome to today's Wolfram live stream. Today we have Willem Nielsen, and he'll be talking about his work working with Stephen on his latest blog post about the fundamental computational underpinnings of medicine. Willem, go ahead.
Thank you, Dugan. And yeah, hi guys. Happy to be here. So I'm just going to share my screen. Okay, so let's go to the top. Okay, so I'm just going to talk about, like Dugan said, about the work that I've been helping Stephen Wolfram with, which is about biomedicine, and it's kind of building on his computational model for evolution, a post that he did this summer. And so before I go into the actual model, I just want to give a high level kind of overview of what we found, which was basically, the takeaway from the biology post is kind of that biology is complicated because of computational irreducibility, in part because so you have these kind of simple rules, but then they just interact and create this endlessly complex organism. And um, so our idea here was to take that complicated organism and see what happens when you try to um, do medicine on it. And Stephen's kind of fundamental idea is that it's like you're a medical observer observing something that's far more complex than you can ever fully you can never fully understand it. And so it's kind of similar to his work in physics in that way. Um, and so, yeah, but just to give a little brief update of what the organisms are. So basically, this is his original post, and basically you just make single bit changes to the rule set of a cellular automaton, and you keep the changes that result in a longer finite lifetime. And the basic point is that you can see just from these pictures that the organisms that you get are already very detailed. And it's not clear why they work. So for example, this one, it's not clear that it's going to terminate, but it does. And so we kind of take those organisms in our posts and look at what happens when we perturb them, what happens when we change a single bit, and can we get them back to the original? Can we classify what happens to them? And those type of questions. So I just want to start off by talking about how we actually got this particular automaton because it's really interesting. So it's a similar setup, but the difference is, like I said, so even in the evolution to get what we want is something that's more robust than the other ones. And so what we do is we just evolve it with perturbation. So this picture here is showing like a row of all the perturbations that were done on a single step in this evolution. And what we do is we
a
do,
couple notches, maybe you're like,
yeah, sure. Let's see. How's that?
good. Can you find a place? Like, okay, it's good.
Okay, so then we take the minimum lifetime instead of just taking the one single lifetime. We take the minimum of all the perturbed lifetimes, and so the point is just that you force the organism to get to have to be able to deal with a different environment and still achieve a goal. And really the hardest part is avoiding cancer. So you have to figure out, the organism has to figure out a way to not, even if the one single cell is changed, to not just completely blow up and go to infinity, which is a fit. So if any one of these is a fitness of infinity, or if it goes infinite, then the fitness is zero. So it has to figure out basically how to terminate in many different environments. And so what's interesting and what Stephen didn't really get a chance to talk about is that the these guys here, they all the organisms that we evolve in this robust environment, they kind of have this similar idea, which is to have what I call like a programmed death. So if you notice, like it's like a purple cap at the end, and basically the effect of that is just it prevents any patterns from escaping and going to infinity. So if we look at all the robust ones we evolve, we can see that like they all kind of have that programmed death. And it's interesting because when you evolve regularly, you actually don't see that as much. So let me show you guys. So this is, okay, I'm just gonna.
Again, these are 1D cellular automata that are all like 3 or 4 or 5 colors, however many colors they are, and then they're all evolving from a single pixel at the top.
Yeah, exactly. So yeah, so it's a 1D. I don't know, should I? Okay, but so for example, these set of organisms are just regularly evolved, and what you can see is they obviously don't have that cap, and also they're kind of more complicated in a way. Um, and and so part of that is because, another reason why the robust ones work is because they have these kind of like big, like you could think of them as like pieces of skin where it's a big purple cap, and if you perturb in that area, then the organism's lifetime will be fine. So for example, if I like perturb this guy at step 90,
Mm-hmm.
or maybe even step 80, so you could see, um, then you can see that like anything in this purple area is not going to affect the lifetime. So it's kind of, you can think of it like a piece of skin or a nail or something where it protects it from perturbations. And these other guys, they don't have that. So if I like, so this is an example of a regularly evolved one, and if I perturb it, um, oops, that was the wrong number. So if I perturb it like at step 100 instead, oh, that's, yeah. Yeah, if I perturb it at step 100, then it just blows up to infinity because it doesn't have that like protective whatever. And so,
How did you find those robust ones?
so that's what I was saying before, which is that we evolved in an environment where we perturb them a bunch.
Okay, it's a search.
Yeah,
Right,
so
and
you're
it actually
searching
takes,
through multiple organisms. So an organism is defined by its like rule set, right? Can you plot out the rule plot for one of these? Is that
yeah,
possible?
sure. Yeah, let's see, where am I? So we go, yeah, so the colors are different, but yeah, that's the rule.
Okay, so
And
this is
yeah.
grayscale for what would be these colors, and
Mhm.
um, and so this is the one for the one that's below there.
Yeah, so this is this guy here,
Mm-hmm.
our main rule. Yeah, but yeah, so we're not changing the rules though. We're just changing a cell in the body, kind of.
Mm-hmm.
And it's also interesting to look at what happens when you change the rules, but we kind of didn't really get into that in this post.
And for this,
Um,
for the cellular
mhm.
automata, so this is like radius 1, and how many colors is it?
It's K equals 4, so 4 colors.
Okay.
Mhm.
And the
And
space of that is like astronomical, right? That's why these numbers are so big.
yeah, so there's a huge amount of rules. And the reason why we use K equals 4 in this one is because they're more like, I find that K equals 4 is a little more plastic and able to do more. Like K equals 3 is a little more rigid because there's not so many rules. So it wouldn't work. In getting what we want is robust rules,
Mm-hmm.
and it's harder to get those in K equals 3. Um, yeah,
Yeah.
that makes
So
sense.
yeah, it does. So what we were seeing earlier was like this purple spot, and so the rule was using that purple to mean something like, oh, this is a space where purple always wins, and there'll be a sort of a tapering off effect for that color as well.
Yeah. Yeah, it's kind of like a big section of purple that, yeah, I mean, I'm confused what you're asking.
Well, just that this whatever this rule is doing is treating purple as this color, this fourth color that it can have a function for, and that function is sort of that the purple color will eat
Hmm.
away at any other color that's like inside of it, and
Yeah.
also it'll like have a finite tapering off, like when the when you see like purple row meeting an edge of white, the white
Hmm.
wins, I guess, and
Right, right, right. Yeah. Yeah, I mean, I don't know how it would, if you had less colors, it probably is harder to do that, but I don't know.
Mhm.
We didn't really test that. Um, but yeah, so when we evolve these robust guys, what's cool, so like I said, they're simpler, and it's kind of an interesting uh, idea, because basically what we're saying is when you have organisms that are dealing with uncertainty and they're solving the same problem in many different situations, they automatically get more simpler and more explainable. Like it's easy to see when this guy is going to die, for example, as opposed to this regular one. It's not clear when he's going to die. So it's kind of like, um, and and many people have said this, but it's kind of like in an uncertain environment, it has the effect of simplifying the organisms a little bit. But that's not to say that they're completely like explainable. Like even in our robust ones, there's pockets of reducibility and pockets of irreducibility, like Stephen said. So that's kind of the background. And then so let me go back to this guy. So now we can look at what happens if we try to heal the organisms. So there's kind of a few steps to medicine. One is just even before we try to heal them is trying to classify all the possible diseases. And so one of the kind of claims that Stephen is making in this post is that there's no in the real world there's no discrete groups of diseases, and that you can't completely categorize all the possible diseases because of irreducibility. Things have just there's many different ways for things to go wrong. So if we look at a feature space plot of all the possible diseases, this is what that looks like. And let me try to increase this. I'll just zoom out for a second. And so, oops, let's see. Yeah, so what we can see is that there is some grouping and there's some like patterns that look similar, but there's no complete clustering. Um, and so it's kind of like saying that like in the textbook we have, there's like ICD-10, which is like a classification of all the possible diseases into different categories. And and so like in the real world though, we would expect that doctors would see all different types of diseases that they didn't see in the textbook, and diseases that are in between two diseases. So that was kind of the first step. And then if you go past that and now we're trying to not just classify the diseases but predict what's going to happen. So we can already see from like just this picture here, if we're just making single cell changes and already we're seeing just like completely different patterns. Um, and so the point is that like even with a small change in the environment, you can have a big change to the organism. So as a medical, as a doctor trying to predict what's going to happen it's not easy at all. So um, this is, yeah, so let's keep going. So one thing we tested is what if we use just the width of the organism as a metric for what it's doing, because one of the like critical pieces about doctors is that they don't have all the information, and so they have to work with these high level metrics. And so the question is, what can you do to predict the outcome of some disease based on some high level metric. So in our case, we use the width. So what that means is in this plot, we're taking the width of all the organism at every single step. And in orange here is the widths of the perturbed organisms, and blue is the original. So we can see already that it's going to be hard just from the width to predict what's going to happen, because it's kind of like chaotic already. And so if we just look at width at a single step even, so like this is width at step 25, and on the y-axis is the lifetime. It's already, we can see that it's not a very predictive measure. So like, as we increase the width, the lifetime does go up a little bit, but it's a noisy trend. And so we can expect to see kind of a similar thing with high level metrics in medicine. And so one question
around small thing.
So that's the original lifetime and width. So basically what that's saying is that because these guys are robust, a lot of times they heal themselves, or they're not super affected by the perturbation. So that's kind of what that clustering is.
Well, wait a minute.
The
It's
size of the circle.
um.
How many appeared at that time?
Well, it says width at step 25. So,
Mhm.
and there's a clustering around width 12, which is approximately half. I'm wondering if
No,
there's sort of an aspect ratio to these that's sort of maintained at a half, maybe. Just a thought.
well, I don't think that's the time. So 25 is the time, and then 12 is the width.
Mm-hmm.
So I'm not sure if that corresponds to an aspect ratio, but I do know that I'm pretty sure that width is the original one. So that's why there's so many there, is
Mm-hmm.
because some of them just don't move, or a lot of them don't move. Yeah.
Where's the perturbation introduced in this? Or does it
It's
vary?
all of them before step 25.
Oh, okay.
All the possible perturbations.
Got it.
Yeah. So this is the distribution version of that, and the blue is the median. So again, we're just showing that it's not very predictive. And so like one question is, what if you have kind of a smarter algorithm? But so this is what that's showing, is a machine learning. In this case, it's using it's a decision tree method, kind of, which is interesting because it's similar to some doctors use that as well. But it's very similar just to the median. And so what this is kind of saying is like, it doesn't matter how smart you are. If you don't have enough information because of your disability, you can't make a accurate prediction. And it relates to something we'll talk about. One of the conclusions that Stephen Wolfram makes is that that if you have like a lot of the problem is just getting the right information and coming up with ways to get that detailed information quickly. And so that's like a potential, vector for improvement in medicine, is just getting better and more information. So that was asking what happens if we predict that width at a single step, but this is asking what if we give it more information, and so we're giving it the width history instead of just the width at a single step, and it's kind of a similar story. But I think, and Nick also helped with this one, and and one of the things that he and Stephen concluded was that you could probably get it to be better with tinkering with the model and the data. But the point is that inevitably there's going to be some irreducibility in the system. And so it doesn't matter how much data you have or how smart you are, it's not clear what will happen. Yeah. Does that make sense?
Yeah. So there was a prediction model that was were there neural nets trained? I'm sorry, I kind of missed that.
Decision tree.
Yeah, so we did a small neural net for this one. I don't know the details because again, Nick did it actually. But
I missed that actually. Okay, so there was a decision tree model and also a neural net model.
yeah, so basically this one is like simple, just feeding it to the built-in function to feeding it to predict. And this one here is Nick's neural net that was it was I can't remember the size, but it was
Can
like
you remember,
small.
Nick?
Yeah, very small. Well, I don't remember either. I'm just looking. It's very small. It's like 3 layers, maybe like 32 neurons wide or something like that. So,
Basic MLP, right?
yeah, it's just MLP, nothing fancy.
Mhm. Yeah, so um, yeah, so so after the question of predicting what's going to happen is the question of actually finding a treatment for those diseases. So that's what we're looking at here. And so at first we just look at single bit changes. In the above picture, let me zoom in. In the above picture is just random changes, just to give you a sense. And then these ones are ones we selected that give it the same lifetime.
So it's a
And
targeted intervention. So there's like an insult and then an intervention, and you're trying to target those interventions to get the length identical to what it would
right,
have been without the original insult.
right. So yeah, so like we're just picking out of all the possible interventions for this disease, we just selected the ones here that got it back to its original lifetime of 101.
What is that search space like? Is that like very, very large search space? Because it's an intervention on top of the insult. So it's like there's a two perturbation search.
Right. So there's 15, he says it here actually, there's 1554 possible second perturbations. Now, if you look at like, if you start looking at multiple perturbations, things get crazy quick. And
Wait, is that 1,554 treatments that get you back to the same place?
no, all possible.
Okay. So are you flipping the single point to one specific color? It seems low. Like I would think it would be a lot larger number, but maybe it's not that complicated.
Well, yeah, it's all possible colors too. So it's what? I mean, there's,
I guess
okay,
there's not that
we're,
many pixels between the start of the insult and the end of the lifetime. And it's just that many pixels within the CA for each color. So I guess it's not that many.
yeah, it's like 88, like maybe 80 steps after, and then the width is like about 10 at each step, 800. And then,
And it's only
yeah,
the
you're
subset
changing,
where you're actually inside the CA. Outside,
right,
because then there wouldn't be any interaction.
exactly, yeah, that too.
Okay.
But yeah, so some of these, like a few of the, or actually in this case, one perturbation actually gets it back to the original lifetime, or sorry, the original pattern with all its kind of details. But most of them lose the actual pattern, but are able to get it back to the same lifetime. So this is kind of saying like, and especially if you intervene late, it's very hard to get back to the original. So, which is interesting because like maybe similar to medicine in that quick interventions are more effective. But yeah, so sometimes like in this case, you get a perfect healing. So on the right is like the differences of the number of different cells as you go down in time, and then on the left is the healing perturbation that we did. Yeah, and sometimes like the organism just heals itself, as we saw with that width graph as well. So these are cases where it heals itself, and sometimes it happens like a local healing. So you could think, yeah, like a low level change doesn't have any effect at all. But then in this case, it's like a low level change has an effect for a while and then eventually converges back to the original pattern. Does that make sense?
Yeah,
So
that's really interesting. Like,
Yeah,
yeah,
so
you can see the scars sort of in that plot on the bottom of what the
right,
difference
right,
is.
right, right.
And so it still gets back to that like blue triangle with like the red stripe and that little bit at the end hanging off.
Right, right. Yeah. Yeah, this is kind of like you have a disease with like, I don't know, with like, yeah, it's basically like a scar. You get a cut and then the details of the organism change, but eventually everything is basically the same.
Do you always like look at these creatures as actual two-dimensional creatures instead of one-dimensional living in time? Because this is just like one-dimensional creatures just growing and then dying,
Yeah.
right?
Yeah.
Yeah.
So
Actually, that was
I
my
really like that.
myself. So thanks for asking, Nick. I was going to ask the same question.
Yeah.
Yeah, I really like that question too, which is like, what do we mean by the dimension going down? And I think Stephen said it well. He's like, we're a bit schizophrenic about what we say these things are. So like, you could think about it like an organism growing, living and dying, like, so it would be time. But I mean, the way I think about it is I think about it like that, but I also think about it as it could just be a process within an organism that's happening. Yeah, does that make sense? Like it could be like an organ, like a subsystem, but it could also be like just like a fruit fly going from its, egg to body. Yeah.
Yeah, makes sense. We can treat them in both ways in a way, right? So, for example, for
Yeah.
that car example, it's not really a scar if you treat it as a one-dimensional creature. So it's kind of completely healed and then lived for a while and died as usual.
Right, right, right. Yeah, yeah. And also, yeah, I think it takes a little bit of like mental flexibility to like work at such a high level, because I mean in the beginning I was like just confused about whether the dimension going down was time, or whether this was just a snapshot.
I mean, it's
But yeah,
definitely some kind of evolution of evolving process, not evolving like biological evolution, but like it's unraveling the progress of this organism's pattern. But those things would probably be more like injuries and not scars, something like that, because injuries can
yes,
heal, scars or something more permanent.
right, 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 right. Yeah, so I guess the better thing to say would be scars are like the guys that get back to the lifetime but don't get back to the same detailed pattern. And then these guys are perfectly healing. So there's no scar.
Yeah. And in a
Um,
way, it also makes sense to say that, like, the earlier you have the perturbation or the disease, the higher effect it has, right? Because the further down the evolution, the time, you kind of have the more complete creature already existing. So if you perturb that creature, it's kind of just nothing happens. It's still gonna die soon, right? Because it's already in that program death
phase.
phase, right?
Yes. And also another thing is like when these guys are evolving, they kind of they start using like irreducible methods, because the only way to get from like a fitness of one to longer is you have to like you kind of use the irreducibility, if that makes sense. So like you said, the beginning is more sensitive. Um, Yeah, and it's also just not just a function of, yeah, it's a function of how they evolve, I think, too.
Yeah.
Can I ask something? So how does this, I don't know, connect or relate to the ontogeny interpretation that was in the, at some point, I mean, Nick and I had a conversation about this in one of the live streams on the biological evolution blog post some time ago. So is that the other interpretation that you had in mind, Nick, or is this connected? And what is your opinion, Willem?
Remind me, what do you mean by that again?
So, I mean, you could see these. I mean, because these are clearly, there's a time direction here, right? Like there's a process, and you know, one could see the whole automaton, right, until it terminates as a process of ontogeny, right? Like the sort of the formation of an organism from say the single cell from the gamete or something, you know.
Yeah.
So I'm wondering, what is the interpretation then about about the interventions and the healing and the scars and all that? Because I mean, it seems more like, you know, there's just an intervention in the ontogeny itself, and it reminds me of Michael Levin's work, of course, because that's exactly, you know, the rules of the automaton aren't changed, right? But like you perform this intervention. So the genetics aren't changed, but you perform the
You change
intervention.
it locally in time. You just have one single event of changing the rule at one particular time step and one particular place, right? You just change
Yeah.
So that's kind of the same interpretation. You just mutated one particular rule at just locally in time once.
Right.
So
Yeah, but I was wondering if it made more sense to interpret this whole thing, like the from beginning to termination, as phenotypes. Like this is the full phenotype, and I was wondering, what is the interpretation of the interventions in that perspective, right?
well, only if you like interpret it as if you already have this grown creature and you perturb it, you kind of have to grow it back from that place in order to so in a way kind of remove all of the layers growing up to this intervention point and then regrow it back from this perturbation in place. So maybe in this.
Interesting. Yeah, fun to
I
think
mean,
about, for sure.
I mean, one thing on this point I think is also that we didn't quite get to touch on, which is that, so like we're changing a single cell and at a single location, but you can also ask like what happens if you change a rule. And usually when you change a rule, it's like going to have a stronger effect. I mean, depending on the rule, but because, you know, it's used repeatedly. So it's kind of like the analogy we were using is like changing a rule is like changing, it's like giving the organism a drug, and so you're like changing the low level molecular kind of process across the entire organism.
So it's like gene therapy. You're changing the genome.
No, but it feels to me that's, I mean, because if I understood correctly, you are, an intervention here is one local change of the state, right?
Yeah.
So then I think the analogy is very strong, right? Because you have the rules being the genome, right? The instructions that push the thing forward, and if those don't change, it's like conventional medicine, right? Like where you have an organism that is living or developing, and you know, you consider the full structure as the organism, the adult organism, so to speak, that then can have a sort of stable function across time until death, right? And so I think the analogy is quite strong there, that you think of initial state is the first cell or something, the gamete or whatever you want to call it. The rules are the genetic code in a way, and then the whole history, right, is the phenotype, is whatever, you know, the adult phenotype. And so if you think of changing the rules is basically changing the DNA, you know, of the organism. But if you change something in the way, it's like doing an intervention. Like, and I think Michael Levin's work is super relevant here. I mean, the analogy, I mean, right? Like Michael Levin showed that without changing anything in the genetics, right, you could make an intervention on the bioelectricity pattern of the developing organism and get very weird stuff, like typically stuff that you would only see when pretty wild mutations happened at the genetic level, right? Like several limbs in different places of the, like organs showing up in different locations in the body and things like that. But the genome had not been touched at all, right?
Right.
So I think that the analogy here is super strong with Michael Levin's work. I mean, not that Michael Levin's work is particularly relevant to understand anything beyond, you know, just the analogy, but I think it really, I think, nails down this interpretation, or in my view, it favors the ontogeny interpretation of this automata, right? That if the rules aren't changed, it feels more like a phenotype that develops from a genotype, and making interventions is a bit like, you know, whatever the development process. If you intervene from outside, I mean, if you don't intervene from outside, it's kind of determines that. That's why similar genetics lead to similar organisms in nature. But if you do something that directly tampers with the process, like changing the bioelectricity pattern in a tadpole or something, then you can dramatically change the output of the ontogeny, right? So, so I mean, again, I don't have strong opinions on this because I'm always coming at this as a fun break from my other stuff, but it definitely seems to me like the ontogeny analogy is very, very strong, right?
Yeah, I agree with that, and I like the fact that you can actually break it down like that. So it's either an accident, maybe it's one perturbation, or it's maybe a continuous perturbation. So it's something like a disease that's underlying cause of issues that happen over time. But that's only on the state. Then you have the rules. The rules are like the same as like the genes or something. So you can have like the set of rules could change over time as well. Or there's an external rule, which is the medicine, and it just comes in selectively, tries to make adaptations. So further research here would be great in terms of like a continued insult that just keeps re-emerging, and then like trying to attack that insult with gene adaptations or medicine over time. And then I think this has a lot of room for the exploration. It's really cool.
Yeah, it's really cool.
Yeah, it's funny you mentioned that. The next picture was us doing multiple interventions and adaptively. So trying basically keeping the interventions that get closer to the original lifetime and seeing how well it does. And it does better, as you would expect, with more chances kind of. And like in this next one, if you slow down time basically and give it the chance to intervene at each successive step, keeping the ones that work in terms of getting closer to the lifetime, then it's more effective that way. So it's like an analogy to the fact, again, that if you have a better interface with the organism and more time and more information, then you can do better with healing it. And also, Carlos, to your point, I wanted to say, I was struggling with the ontogeny interpretation when we were just looking at the previous post, with these guys. And part of my issue was just that I just felt like there was no, one of the kind of defining principles to me for an organism is that it has phases, in that it like grows and then it stops growing. And to me, this just felt like randomness. So, but it's nice. It was like nice for me to see when you give it, when you throw in some uncertainty, that it does seem to have that like growth and death phase. And then, so then in that case, the ontogeny interpretation made felt better to me. And so yeah.
Mhm. Yeah. Yeah. What I was thinking is it might be fun to consider, if you put, because this feels like ontogeny, but living beings are agents, right? I go back to I mean, it wasn't really criticism, but my comment back then when I was saying, you know, can we really say that these things are organisms is if we are just like looking at some like width or depth or whatever of or like some random parameter as fitness, like we said last time, right?
Mhm.
But I find it interesting that if you think of the ontogeny of an agent, right? Like, and this somehow defines something that is agential, which in my view just corresponds to putting many of these things in a network to interact somehow, right?
Mhm.
And then I don't know, do you maybe you put the automata themselves in a network to interact, and like the rules maybe might be like interacting, or you just take the output of each automata and somehow define a very simplistic agent from those,
Mhm.
and then you put those in interaction, right?
Mhm.
And so with that perspective, you can start to like do evolution, like actual evolution, in the sense that you can get different genetics that you begin with random things, right? And you define some sense of competition, like maybe there's like a resource and there's like a predatory behavior or something, right?
Mhm.
And so you can do very simplistic like network model of maybe there's like resources distributed in the network, and there's agents that eat other agents or something like that, when they are
Mhm.
larger or sufficiently larger or whatever, something very simplistic,
Mhm.
but could be fun to just like use these things like these automata as actual genetic code for
Mhm.
otherwise agents that are in interaction. Like the way I'm imagining it is very like you have agents on that are super simple, right? Like they have some discretized space to move around. There's some discretized resource like food, and some
Mhm.
meaningful like cross section. When two come together, they can either just move past each other or they can eat each other or something like that, right?
Yeah.
And maybe if you sort of couple these two systems, like you have the automata part to basically give you a pool of genetics, and you have these things constantly propping and having some sense of descendancy, right? Like where they are like parents of their children and so on, and like that way you can have a natural selection mechanism, like a very simplistic natural selection mechanism. I think it would be cool because for me, the fact that you have automata like terminating automata as an analogy for a like for ontogeny, and you have an organism that is sort of formed after a process, to
Mhm.
me seems powerful enough that you can say, well, because there are so many of these already, I mean finitely many, but many, right? You can just have a bunch of them in some kind of population, and you
Mhm.
introduce some really minimalistic uh, analogous elements like competition or food seeking or predation or
Mhm.
whatever, and
Mhm.
maybe you just infuse life into the system, and you actually get evolution, and you get very very refined genotypes by the end of it, which is like, oh yeah, this thing is clearly the biggest possible thing, because if you define like whatever some parameter to measure when something eats something, maybe you just optimize for that genome, and at the end of the process you actually find that everything is like as large as possible or something like that, like
Mhm.
I don't know,
Yeah.
just fun idea. I have no idea what I'm talking about. So
No,
Yeah,
I
I
think
think
it's a
that
good
was
idea.
something that was looked into was you have like this organism with its genotype and this organism with that genotype. And let's say you're trying to make organisms that have a finite lifetime, and you want to optimize that lifetime to make it the longest finite lifetime.
yeah.
And so you have like all these randomly selected organisms, and you try to merge two good ones to get like a genotype on like a third one that has like the best of both features, let's say. But I think
Yeah, I mean,
that
the
was
next level would be to introduce sexual reproduction, as you
oh,
said.
that's what I'm saying. Is like you want
Yeah, yeah.
this one, and they're prime candidates. So you want like something that takes the best from both genotypes. And
Yeah.
I'm not sure how successful that was on the metric of these longer lifetimes is what you're optimizing for as a fitness function.
Right, right.
But
Yeah,
you're right.
but that's why I mentioned back then that you need some form of like I believe you need a minimum competitive system or like something that can implement some form of natural selection, and so you need some kind of network of interaction between these things. Like they can't just, I mean, you can have some preliminary conclusions, but I I wouldn't take it too seriously until you put things in a network of interaction. I mean, because that's what an ecosystem is. In my view, that's what life is about. You have things in a network of interaction, and it really is about interacting sort of pockets of computation like those, right? Like life has tended to be this sort of like nucleation of computation in a sense, right? Like your RNAs or your peptides or whatever like tended to sort of clump together and do their thing, and then they interact with others and so on. And obviously, we all regard like Luca as like the breakthrough, right, in the history of biology, and like this single cell that was already a single cell. Like at the moment you have a lipidic layer that is separating the inside from the outside. Like that's a lot of biologists recognize that as the most important moment of like, okay, yeah, life really got going at that point, right? So to me, that is just another very strong expression of things being clearly in pockets of interaction, right? These things are going to be separate from their environment and separate from each other, and the interactions then happen in this pocketed ways, right? So if we don't, this is all fascinating, obviously. I'm not saying there's anything wrong with this, but until I think we put these things into a network somehow that interact, I don't think we're gonna see a lot of like biology phenomena proper.
Yeah, I think
the minimal. Can I just like a quick idea? The minimal model, the minimal model, you don't have to actually put them into network because you already can treat this genotype also encoding some rules of interaction. So, for example, in the previous block about biological evolution, we looked at the different initial conditions. Basically, you start not from a single cell, but from multiple cells, and now we can treat them as multiple organisms evolving with like a single environment and interacting. So, in a way, the genotype is not just encoding a single growth of the individual, but also their interaction. So in a way, this also lifetime now encodes not just life of the single individual, but their like the whole environment, their cooperative lifetime, so to speak.
Yeah, I mean, there's a lot said, but well, to Carlos's point, I mean, I definitely agree with you that it's unsatisfying to have. I feel like one thing we're doing is we're putting in agency from the top, meaning like the agency isn't emerging. So we're saying that these organisms automatically want to live longer finite lifetimes, and it would be nice, I agree, to have that emerge where you just kind of have this soup, and then like through scarce resources you should see that agency, or like the things that try to survive and figure out and become intelligent in a way are the ones that survive. And I'm actually looking for something. I did something on this, but the other point I wanted to say was I just feel like I think I kind of agree with Nick's point on this, which is that like if it's not like the model is never going to represent the entire thing, and I think it's better to try to be a little flexible with a simple model rather than try to throw everything into the model. So like we could, I agree that it should be interesting to try the interaction, but I'm skeptical that, you know, it's going to be really hard to like to do it. And also, yeah.
Yeah, for sure. I think Brian, at some point you wanted
Yeah,
to.
I also think what Carlos is driving at is the kind of the Conway's game of life nature, so that the systems can interact with each other, and I think that would be like an interesting approach as well.
Yeah, yeah. No, and I take your point, Willem. I'm not suggesting that, you know, this is somehow flawed because you're not putting them in interaction or anything like that. I think it's definitely super interesting to do it this way. But because I mean, I was motivated by the strong analogy with ontogeny, right? Like it's more like try to somehow what I'm trying to say is it's more like you try to exploit as much as possible that interpretation because it's very, very strong, right? Like to me, it is as strong as it gets as a model, right? Like obviously you're not modeling genetics as we know it because you're not modeling like, you know, the bases and like the transcription or anything like that, but you are very, very close to what nature seems to be doing, right? Like you have some finite states within the range of energies that, you know, the chemistry, the biochemistry makes sense, and you have some rules that are defined by these discrete states, and then you are letting them evolve, and they have exactly this behavior, right? Like they have the behavior that they terminate, and they have like they're combinatorially bounded. And so to me, it's like a very strong analogy. It's not, by no means I'm trying to say that you know, other ways are wrong or worse in some sense, but to me, the fact that you have captured so powerfully one of the aspects of biology, which is this ontogeny, with the analogy with the phenotype and genotype and so on, to me, is my intuition is like, okay, let's try to exploit it as much as possible. But I get the point. I get that you can't see like the one-dimensional space of automata as the environment, and you can see the states as the organisms interacting. Of course, I mean,
Yeah.
I totally agree that that's definitely relevant. But I think the other thing that would be interesting there is to do a bit of like observer theory like analysis and identify, you know, the notion of individual in those places, because and there's some literature from what's his name, David Krakauer, who's done some theoretical work on the notion of an individual, and as far as I understood, it was quite operational. So it might be possible to actually extract some of the notions of what they define as an individual, because of course, as you understand, you know, they were applying this to anywhere from like organelles to cells to genomes to populations to microorganisms to colonies to, you know, all these kinds of things across biology, and maybe other media or other substrates as well. So anyway, I think it would be interesting, because the question of my only point about treating the whole state of an automaton as a community in interaction via the rewrite rules is that I like the notion of individual there is a bit more subtle, right? It's not so clear. Like you clearly have a system of interacting things, sure. I mean, that we agree on, but it's less obvious where you can say, hey, this thing is now an alive organism, and this is then interacting with this other alive organism, right? That is more contentious to interpret, and it probably requires some further analysis or reduction or something, right? Because I mean, I don't know if either you, Willem, or Nick, you have a clear picture of what the individual means in those settings where you have the whole state of the automaton as the organism, as the population. Sorry.
Well, you have this clear quiescent color, which is assumed white, right? So it kind of acts as a separator already. So you can just
Okay.
look at one line state and whatever, how many
So
those
adjacent colored
continuous color.
cells are one organism, right?
Yeah,
Okay.
just one dimensional organism. So you can basically count organism at each time step. How many of them are there?
Yeah, that's a well-defined first notion. That's a good answer. Yeah.
Yeah, I mean,
But you have rules doing
relating
weird
to,
things, right? Because you have rules that where organisms like split up and join up and like they do weird things, right? But
Yeah.
yeah. But yeah, relating to the interaction thing and also the sexual reproduction thing is, so in the original post, Wolfram, or yeah, you guys tried to do recombination of the rules, and it kind of didn't. The result was that it worked the same as mutation basically, and so I mean I've been trying different things to try to have a model for interaction, have a model for recombination, and so one of the things I've been trying is this, which is to instead of recombining the rules, you just recombine the initial conditions. So I mean, it's obviously it's a little messy, but what this is showing is basically it's an evolution, but you're scrambling the initial condition into different parts, and so the organism kind of has to develop modular parts, and so it's kind of similar to what we were doing in the medicine post, but instead of doing a single bit change, it's like you just rearrange the initial conditions, and the hope is that like from that you see the modular parts, and then also you would see that recombination does better than mutation in this case. But I'm not sure if it's going to work like that.
that's interesting.
I'm not sure what do
And
you
then,
mean by recombination of initial conditions here. Just have a single one, right? Or you're actively doing something.
let me see if I can find a better picture. Oh yeah, like so if you look here, so in this case I split it into 4, and then I'm just arranging those 4 pieces. Does that make sense? So like this picture, and each row is a possible arrangement of 4 different pieces. And then this is the result of those, what the lifetime you get when you do those rearrangements.
Yeah, so you're mutating and also recombining. So instead of treating a rule as a bit string and trying to mutate it or recombine it, you just treat the initial condition and do the same kind of things there. And you're saying
Exactly, yeah.
recombination works better than mutating.
Well, it hasn't worked better yet, but that's what I'm trying to get.
Okay.
Yeah, but I forgot what I was going to say. Oh yeah, I mean, I also think that there's room, like, back to what Carlos was saying about the Michael Levin thing, which is that, like, if you think about the genes as the rules, and then the body or the cells as the state, I feel like that's also a little bit misleading in that it doesn't leave room for the agency of the cells and the other things other than the genes, and that's kind of like a big part of what I think Michael Levin is saying, which is that the other parts have agency too. And so I think you can also flip it around, and I mean Stephen Wolfram did this and just say that and like Nick was saying, which is the initial conditions are the genome, and then the rules are the body, and think about it that way. And in that sense, like the genes are just kind of like a memory bank, and the parts are doing the work in a way. But yeah, I don't know if that's like fruitful.
Ryan, did you have a question earlier?
No, no question.
Okay.
Yes, I just wanted to get back to the rest of explaining this. So one cool thing, so yeah, we were talking about just therapy and doing quick interventions, like what James was saying. And so a kind of an extension of that idea is to try to predict where you should do a therapy using machine learning based on some observed metrics. So in this case, we give the machine learning algorithm the widths, and we ask it to predict where to give a therapy. And so in this case, the red arrow is where it chose the therapy. And so, yeah, it's like a similar, so the thing is, it actually doesn't work that well. It looks like it's working because the organism, as we mentioned, it heals itself a lot. So what this is the lifetime of the original divided by, or the lifetime of the machine learning treated over the regular. And so you get this, it just looks very flat, and like the machine learning thing isn't working that well so like the lifetimes are just all over the place basically. And this is the last section, which is that, and this is definitely relevant to what you guys were saying, which is that you can also think about doing like a computational version of a clinical trial. So like so in this rule set, this is the rule set for our original main rule that we've been looking at, and the 3 yellowed highlights are ones that aren't used. So basically you can change those and get an organism that's exactly the same, but with a different genome. So it's kind of like it's like an organism that has the same phenotype but a different genotype. And so we're kind of using that to say, so like when the clinical trial happens and everyone has the same symptoms, but I mean, obviously their phenotypes are different, but we're kind of saying, what if just the genotype is different, or just the underlying rules are different, but the pattern is the same? What happens when you change the organism? And what you see is basically that there's just a lot of variation for what can happen when you have different genotypes. So this is the kind of the spread of all the possibilities for all the different genotypes of what happens when we do that perturbation with the gray arrow at the top. And so that's like kind of the spread of diseases. But then we can also look at the spread. What happens if we give them all the same disease and then do like a clinical trial, which is you give everyone with the same disease the same drug, and then you try to, from that, interpret like you try to say whether the drug is effective. And the point here is just that there's just like a ton of variation when you have different genomes, and it's very hard to say based on a subset of that genetic population whether that will kind of expand to the whole population, if that makes sense.
What's changing here in each successive plot? Is the genotype changing? The rules?
Yeah. So these are all different genotypes. So I think there's 64. Yeah, let's see. Yeah, 64 different genotypes. Because there's 3 rules that are, so it's just all the combinations of those 3 rules gives us 64 different, phenotypically similar, genetically different organisms.
Yeah, so it's 4 colors, so 4 cubed is 64. And then
Mhm.
why are those highlighted? Is that because, I mean, the phenotypes are the same up until like both the interventions are implemented.
They're highlighted because those are basically like genes that were not expressed.
Right. That's okay.
Yeah, that's the way to say it is inactive genes.
When you change them, they have the possibility of suddenly becoming expressive. So it's like some latent gene expression.
Yeah.
Mhm.
Right. Exactly. Yeah. So that's kind of it.
Are you perturbing those like specifically so that this particular gene rule in the genotype activates, right? For that particular one of those 64.
Okay, good question. So not so they all, well, let's see. So in some of the cases, the disease in the original rule and the disease in the genetically different rules are the same because of what you're saying, which is that the rule never gets activated even with the disease. And that's this case here without the red arrow. So the red arrow is showing the first time that the new rule is used. So up until that point, it's exactly the same. And then basically that's when the new gene gets activated or gets used. And so then things go haywire.
So why does it get used if nothing changes before?
So basically, okay, so in this original pattern, where is it? In the original pattern we had, it's probably down here, it's closest. Yeah, in this original pattern, the way the rules evolved, that one never got used, right? Those three never got used. But when we give it a disease, that's not necessarily true anymore. So then we get this new pattern, and then because of the new pattern, that new rule sometimes gets used. Does that make sense?
Yeah. Only sometimes when there's no red arrow, basically that genotype still evolved as the original, right?
Mm-hmm.
Not
Yeah.
using those rules. Yeah. Okay.
But yeah. So that's the post. Thanks for listening.
Very good stuff, Willem.
Thank you.
Yeah.
Yeah, that was
Thank you
great.
so much. Really great
What I
and
like
great
about
work
it-
on the blog. Sorry, James. Go ahead.
There's a great direction here for the investigation as well. I think this is kind of like just starting to open the lid on what is biology and medicine. There's a lot here. This is going to be a very fruitful area of research for sure.
Mm-hmm. Yeah, I 100% agree. And I think definitely the ideas that you guys were talking about are all sound interesting to me, and I'm motivated now to work on it. So thank you.
Amazing.
Great. Yeah, I have a couple of community announcements. Maybe I can just let everyone know about that. If you want to stop sharing from this. Okay, great. So firstly, okay, so tomorrow we have another live stream with Rocco, and then Saturday we're having a community forum. So anybody that would like to just come and chat and hang out, please join us 2 p.m. Eastern on the Discord server. The theme for this community forum is philosophy. So we get all philosophical and discuss stuff as rigorous or unscientific as you want. It's going to be just a fun time. Then also, as you might have noticed, we are going to very soon be having merch. So yeah, stay tuned for that. Swag store will be launching next week, and this is going to be a limited edition first drop. So as soon as that merch goes up, you would want to grab that as soon as possible. Yeah, so that's it from us. See you guys tomorrow, and see you on Saturday.
Bye, everyone.